Hello, my name is Wynter Rentas, and I'm one of this year's Eureka Grant recipients. I'm here presenting my senior project titled Antimicrobial Properties of Dog Saliva. So a little background about my project this year is based off a study according to Bowen 1974, which basically analyzes the different properties in saliva. Saliva has been referred to have contain various antimicrobial substances, such as lactoferrin, leukocytes, lactoperoxide, antibodies, and cationic proteins. In order to test this hypothesis, studies have been done to test to see if dog saliva does in fact possess antimicrobial properties. Another study was done to address the question of whether saliva was bactericidal for certain wound contaminants. This specific study, Antimicrobial Properties of Saliva, Role in Maternal Perpetuant Grooming and Licking in Wounds, found that dog saliva does possess antimicrobial effects against Escherichia coli and Streptococcus canis. This was found to be true in relation to dogs giving birth and the adaptive behavior they demonstrated. This was when they licked their mammary and anogenital areas. The study also verified that dog saliva was not effective on the bacteria Staphylococcus and Pseudomonas aeruginosa. The authors hypothesized those bacterial species are biologically insignificant to the dog's health. Due to the saliva not being able to kill the bacteria Staphylococcus, it may be a reason why it is oftentimes present in dog wounds. Further research is needed to confirm whether or not dog saliva does possess antimicrobial properties and which bacteria in specific are most affected by these properties. So the topic of my senior research project is basically to see if dog saliva does contain antimicrobial properties. Growing up, I always heard that canines' mouths were said to be cleaner than the average human, but I never understood how. My experiment goes to address my theory that dog saliva does possess antimicrobial properties. The importance of my experiment is to prove that dog saliva does contain antimicrobial properties, which in the end potentially helps to aid in the maintaining of the canine's health. My hypothesis is that there will be a decrease in bacterial load once Enterococcus faecalis, Pseudomonas aeruginosa, and Staphylococcus aureus are exposed to canine saliva. The materials and methods that I used were I selected 5 canine recipients. From those canine recipients, I collected saliva samples and froze it until I was ready to use them. After that, I rehydrated 3 different bacterial samples, as mentioned earlier, Staphylococcus aureus, Pseudomonas aeruginosa, and Enterococcus faecalis. I then used brain heart infused agar as the medium to hydrate my plates. This type of agar was chosen because it works best for growing different types of strains of bacteria. I then exposed the 5 saliva samples to the 3 bacterial samples. This was done by streaking the bacterial samples across the entirety of the Petri plates. And then once that was done, 1 assay disc from each saliva sample was placed onto the plate in addition to the control assay disc. The plates were then incubated for 24 to 48 hours before results were obtained. The incubation period was varied depending on the type of bacteria. Petri plates after were then analyzed to see if a decrease in bacterial load was present due to any of the saliva samples. After that, observations and data were collected as part of the results. Here I have a table of the 5 canine recipients that I used. Here we have the age, weight, sex, intact or neuter, spay, breed, and the grade of the dental disease. In the next following slides, I will show a picture of each canine recipient and tell a little bit about them. Our first canine recipient is Ruble. Ruble is a 7-year-old, 98-pound, male neuter Borzo with a grade 1 dental disease. Recipient 2 is Geronimo. Geronimo is the 11-year-old Borzo, 70-pound male neuter with grade 1 dental disease. Recipient 3 is Talois. Talois is a Chinese crested, 14-year-old, 7-pound male neutered with grade 1 dental disease. Canine recipient 4 is Otzi. Otzi is a Chinese crested, 3-year-old, 16-pound intact male with grade 1 dental disease. And recipient number 5 is Flash. Flash is a Chinese crested who's 12 years old, 16 pounds, male neutered, and also a grade 1 dental disease. So the procedure of my experiment is as follows. My study aimed to prove that canine saliva does possess antimicrobial properties. This was done by exposing 6 different canine samples to 3 different bacterial samples. These plates were then analyzed over 3 trials to see if the exposed saliva samples made a decrease in the bacterial load on the plates. If positive results were found and there was growth inhibition present, then this data would be useful for veterinary clinics to understand why dogs always want to lick their wounds, opposed to getting hurt or giving birth or after surgery. Finding the bacteria in which canine saliva is antimicrobial against will help to determine possible ways in the future in which canines can heal themselves. Here, we're going to talk about some of the results that I found throughout my experiment. This is trial 1, and table 1 displays that in trial 1, none of the plates for neither of the 3 bacteriums showed a decrease in bacterial load once exposed to 5 saliva samples in comparison to the control disc, which was made up of saline solution. Each plate consisted of 5 assay discs, 1 from each canine recipient, and the control plates each consisted of 5 assay discs that were soaked in saline solution. Here, I have a picture for trial 1 of the bacteria Enterococcus faecalis. Enterococcus faecalis is a common bacteria found in the GI tract, on the skin, and in the oral cavities of dogs. Here you can see that the bacteria was not streaked over the entirety of the plates, so there was less surface for the bacteria to grow. In response to the saliva not killing any of the bacteria, you can see in one of the plates on saliva sample 4, to the picture on your right, the second plate, where the bacteria completely saturated the assay disc. Trial number 1 of Pseudomonas aeruginosa. Pseudomonas aeruginosa is a pathogen that is typically found to lead to second infections. The most common infection caused by this bacteria is an ear infection in dogs. When analyzing, out of all the bacterial cultures, I found these plates to be the least colonized out of all 3 bacterial species. The reason for this is unknown because based off the way in which inhibition of growth was determined in my experiment, there was none present by any of these saliva samples. The saliva samples on this plate were also not saturated nor touched by bacteria like the other plates. We see here bacteria colonies are only present throughout the plate but not present on the saliva samples. So on the left side we can see the white dots on the plates are the bacterial colonies, but they didn't form and congregate like in trial number 1 with Enterococcus faecalis. And these are my plates from Staphylococcus aureus. Staphylococcus aureus, also referred to as staph infection in humans. This bacteria causes infections in dogs. It's typically developed in a dog in response to any type of allergic skin reaction or disease. This bacteria typically attacks the outside skin surface of dogs and causes bumps, redness, and irritation. What really stood out to me was that in these plates, we see that the bacterial colonies formed outside the exterior of the circle versus directly on the saliva samples. The reason for this is unknown because this experiment was only testing inhibition of growth, which on these plates, there also was none found. Another interesting observation of this bacteria sample was found on plate number 3, which is towards your bottom right. in which we see here a vast majority of the bacterial sample covering the whole plate, but at a more concentrated setting. If you look close attention to the bottom part of the plate, you can see a dark tint around saliva sample number 5. These are the results from trial 2. Table 2 displays that in trial 2, none of the plates for neither of the 3 bacteriums showed a decrease in bacterial load once exposed to the 5 saliva samples in comparison to the control. So trial 2, Enterococcus faecalis plates. Trial 2 exhibits multiple discs for this bacterial species that were saturated in bacteria versus the control. So to the left, you have your 3 plates with the saliva samples on them, and then to your right, you have the control. And if you can take your attention to the left pictures, you see that there's 6 assay discs that are saturated and covered in brown bacterial species. In trial 2 of the plates of Pseudomonas aeruginosa, we see that there remains no inhibition of growth nor growth patterns of bacteria. The saliva samples stayed relatively clear of the bacteria, which was interesting to me because in trial 1, you see the same results. You see the same growth pattern, and you see that none of the assay discs that were filled with saliva samples were at all touched by bacteria. In trial 2 of Staphylococcus aureus, we see that the growth patterns remain the same as was present in trial 1, drawing special attention again to the control plate, in which we do see slight inhibition of growth within each control disc. So paying close attention to the left top plate, labeled control, you can see that the assay discs that are filled with saline solution, we can see that there is slight inhibition of growth across the plate. You'll see that it's exhibited by a clear ring around the disc, which is present only in the control plate. Trial 3. Table 3 here shows that in trial 3, none of the plates for neither of the 3 bacteriums showed a decrease in bacterial load once exposed to 5 saliva samples in comparison to the control. Trial 3 here, Enterococcus faecalis plates. Observations remain the same as the previous trials in regards to the growth patterns of bacteria in the absence of inhibition of growth from the saliva samples. So for the next 2 slides, I will show you again just plates from trial 3, which replicated basically the results we've seen in trial 2. Here in trial 3, we have the Pseudomonas aeruginosa plates. And for trial 3 plates of Staphylococcus aureus, we have our 3 plates in the control. So a little bit to discuss about my project. Variables that could have altered positive results for showing inhibition of growth could have been that for multiple reasons, the species that I chose for my bacteria are not typically species found in dogs. So that could have been a reason why the saliva samples didn't show inhibition of growth once exposed to these bacteria, is because the bacteria that I chose are not typically found in dog wounds or on dogs' skin or in their wounds. Also, the saliva to bacteria ratio could have altered the results. The bacteria samples outweighed the amount of saliva present. So I kind of thought about it and looked at it in the way that the amount of bacteria I had was consistent throughout the whole experiment, yet the amount of saliva I had, which covered an 8-millimeter disc, was disproportionate to the amount of saliva that I exposed the plate to. Another variable could have also been the technique I used to collect the saliva samples. It could have not been as efficient in collecting the adequate amount on the assay disc. So the way in which I collected the saliva samples was by using a glove and sticking my hand in the dog's mouth to saturate the whole disc under the tongue. That could have made it harder for me to collect the amount of saliva that I needed in order to get positive results. None of the questions that were addressed early in the study had any effect on the results found in the experiment. No saliva samples showed to inhibit the growth of the bacteria. Some plates displayed more bacterial load, and this was due to the way the bacteria colonized and had no correlation to the properties of the saliva samples. If this experiment was to be redone for further studies, I would include various pathogens, also known as bacteria, with a focus on using pathogens that are specifically found in dogs' wounds, which are Escherichia coli and Streptococcus canis. These pathogens are both heavily associated in neonatal septicemia of canines and was used in the experiment by Hart and Powell. As well, I would also consider as immediately exposing the saliva samples to pathogens following sample collection and not freezing them. In my experiment, what I did was I exposed it and I froze them for 2 weeks until I was able to use them for plating. As a result of freezing them, I feel as if it could have affected the enzyme proteins found in the canine saliva, making the saliva not as effective in antimicrobial properties. Even though I was unable to show inhibition of growth in my experiment did uphold Hart and Powell's concluded study that dog saliva is not antimicrobial against the following bacteria: Staphylococcus and Pseudomonas aeruginosa, which were 2 of the bacteria specimens that I used in my experiment. Hart and Powell came to that conclusion because they found that those bacteria are biologically insignificant to a dog's health. So in other words, these bacteria are not typically found in dogs. So there's no way to tell that they would be antimicrobial against them. Here are a few acknowledgments that I would like to give. I would like to thank the Eureka community for choosing my project as one of the grant recipients. My advisor, Francisca Mendoza Herculade, for helping me throughout this entire project. My mentor, Crystal Cox, for helping revise my work and supporting me throughout the entirety of this research project. My professor, Rachel Messer, for believing in my project and providing moral support. And the amazing Dr. Burks, not only did she provide me with her expertise as a veterinarian, but she also let me use her 5 amazing dogs. I want to give special credit to my research assistants, Capri Bison, Brianna Reeves, Hannah Watts, and Shelly Clark. I could not have done any of this without each and every one of your help and support throughout this research project this year. Here's a little bit of my lit review, and I would like to say thank you for watching.